Hello, my name is Michelle King, and my presentation will be on the Polygene Hypothesis and the statistical tools for quantitative inheritance. So let's talk about the Polygene Hypothesis. So this is an idea in the field of quantitative genetics that proposes that multiple genes, each with small individual effects, collectively contribute to the inheritance of quantitative traits or complex traits. These traits are typically controlled by multiple genetic factors and are influenced by both genetic and environmental factors. The polygene hypothesis contrasts with Mendelian genetics, where traits are controlled by a single gene with a dominant or recessive effect. So, let's talk about multiple genes. So, according to this hypothesis, quantitative traits are controlled by the action of multiple genes, often referred to as polygenes or quantitative traits, QTLs. Each of these genes have a small effect on the overall phenotype of the trait. Let's move on to continuous variation. Traits subject to polygenic inheritance typically show continuous variation in the population. This means that individuals exhibit a wide range of phenotypic values, and the trait is not easily categorized into distinct categories. For example, tall versus short, as in Mendelian traits. Now let's move on to additive effects. In the polygene model, the effects of individual genes are additive. This means that the contribution of each gene to the trait's phenotype can be summed to determine the overall phenotype. For example, if there are 3 genes contributing to height, the height of an individual can be understood as the sum of the effects of those 3 genes. Lastly, let's talk about normal distribution. When many genes with small effects are involved, the traits often follow a normal distribution or bell-shaped curve in the population. This distribution results from the cumulative effects of multiple genes and environmental genes. Let's continue talking about the different aspects of the polygene hypothesis. So let's touch on environmental influence. While genes play a significant role in determining the trait, environmental factors can also influence the phenotype. These environmental factors can include nutrition, temperature, and other external influences. Quantitative traits. So, polygenic inheritance is commonly associated with complex quantitative traits such as height, weight, intelligence, and various diseases like diabetes, where the phenotype is influenced by many genetic and environmental factors. Let's talk about the polygenic score. Researchers often use statistical methods and genetic markers to estimate an individual's genetic predisposition for a complex trait or disease based on their polygenic score, which takes into account the collective effects of multiple genes. Lastly, let's talk about empirical support within the polygene hypothesis. So, the polygene hypothesis is supported by extensive empirical evidence in agriculture and human genetics, aided by advances in genomics and statistical genetics. The polygene hypothesis has been supported by a wealth of empirical evidence in fields like agriculture, crop breeding, and animal husbandry, and human genetics, studies of complex diseases and traits. Advances in genomics and statistical genetics have enabled researchers to identify QTLs associated with many complex traits, providing further support for the polygene hypothesis. So let's dive into the world of statistical tools. So, unlike Mendelian genetics, where we deal with simple traits controlled by one or few genes, quantitative genetics focuses on complex traits influenced by multiple genes. So, to unravel these mysteries of these complex traits, we rely on statistical tools that help us make sense of the data and uncover the genetic and environmental influences. So, let's talk about some of these key statistical tools. We have heritability analysis, which is a fundamental concept in quantitative genetics. It quantifies the proportion of the variation in a trait that can be attributed to genetic factors. To estimate the heritability, we use tools like twin studies, family studies, and statistical models. This helps us understand how much of a trait's variation is due to genetic inheritance. Let's talk about regression analysis. Regression analysis is a powerful tool in quantitative genetics. It allows us to access the relationships between multiple variables, such as the influence of a specific gene on a trait. By analyzing large data sets, we can identify genes that contribute to complex traits and their individual effects. Let's move to GWAS, Genome-Wide Association Studies. This is a cutting-edge approach that involves scanning the entire genome to identify genetic variations associated with these complex traits. With the advent of high-throughput genotyping technology, GWAS has revolutionized our ability to pinpoint genetic markers linked to diseases and quantitative traits. So let's keep it pushing. We have quantitative trait loci, QTL mapping next. So QTL mapping helps us locate specific regions on chromosomes that are associated with these traits. By examining the inheritance patterns of these traits across generations, we can narrow down the genomic regions where the contributing genes can be found. Lastly, let's talk about the polygenic score calculation. So, polygenic scores are derived from a combination of genetic markers associated with a trait. These scores allow us to estimate an individual's genetic predisposition to complex traits like obesity, diabetes, or even how intelligent they are. Statistical algorithms play a crucial role in calculating these scores. So these statistical tools in quantitative genetics have diverse applications. We use them in agriculture to breed crops with desired traits, in medicine to understand the genetic bias of complex diseases, and in evolutionary biology to explore the genetic basis of adaptive traits. By employing these tools, we gain valuable insights into the interplay between genetics and the environment in shaping complex traits. So, to summarize, statistical tools in quantitative genetics are indispensable for unraveling the mysteries of complex traits. They allow us to estimate heritability, identify genetic markers, and map the genomic regions responsible for these traits. These tools empower us to better understand the genetic and environmental factors that shape the diversity of life. So now let's talk about our broad sense heritability, often denoted as H squared. This is a measure used in quantitative genetics to assess the proportion of the total phenotypic variance in a trait within a population that can be attributed to genetic variation, specifically including both additive gene effects and non-additive gene effects. It provides insight into the overall genetic influence on a trait's variability. In the context of broad heredity, we are interested in understanding how much of a variation in a particular trait is due to genetic differences among individuals in a population. This includes both the additive effects of multiple genes, polygenes, and any interactions between these genes. Heritability is typically expressed as a value between 0 and 1, with higher values indicating the genetic factors play a significant role in the trait variation. So here we can see the calculation. We're given the equation where H squared is the broad sense variability and our different terms to substitute into. A high H squared value close to 1 indicates that a significant proportion of the trait's variability is due to genetic factors, both additive and non. A low H squared value, closer to 0, suggests that the trait is primarily influenced by environmental factors and has little genetic contribution. So I've included an example of broad heredity. So let's say researchers study human height in a population and find that the phenotypic variance in height is 100. After conducting genetic analysis, they determine that the genetic variance, which includes both additive and non-additive genetic effects, is 80. So then we'll use the formula to calculate H squared. So in this example, broad sense heritability h² for human height is 0.8, which means that 80% of the variability in height within this population is attributed to genetic factors, including both additive and non-additive genetic effects. So let's cover narrow heredity, family or narrow sense heritability. So narrow sense, often denoted as lowercase h², is a measure used in quantitative genetics to assess the proportion of the total phenotypic variance in a trait within a population that can be attributed to additive genetic effects on the individual genes. It focuses specifically on the contribution of these additive genes to trait variability and excludes non-additive genetic effects and environmental factors. Narrow sense heritability provides insights into the genetic basis of the trait's variability. So here we have the calculation along with what we need. So here the h² can be interpreted as when there's a high h² value close to 1, it indicates that a significant proportion of the trait's variability is due to additive genetic effects. If there is a low h² value, it suggests that the trait is primarily influenced by non-additive genetic effects and environmental factors, with little contribution from the additive genetic effects of the individual genes. So let's take a look at this example. So let's consider a population of pea plants with variations in flower color. Researchers study the total phenotypic variance in the flower color and find it to be 100. So after conducting genetic analysis, they estimate that the additive variance, which represents the variance due to the additive genetic effects of a specific gene, is 70. So then we'll use the formula to calculate. So in this example, narrow sense heritability h² for flower color in pea plants is 0.7, indicated that 70% of the variation in flower color within this population is attributed to the additive genetic effects of individual genes. Estimating the response to selection in genetics is a critical step in breeding programs aimed at improving specific traits in a population. The response to selection quantifies how much the mean value of a trait change in a population due to the selective breeding over one or more generations. Here's how you can estimate the response to selection. So number 1, you'd have to define your objective. This means to determine the trait or traits you want to improve through selective breeding. You should have a clear understanding of what you aim to achieve in this step. Next, we would have to collect our data. So gather data on the trait of the interest from the starting population, often referred to as the base or initial population. This data includes phenotypic measurements such as height, weight, and different things like milk yield. And if available, also the genetic information, so like pedigree or genomic data. Third, we would like to select some candidates. So you'd have to identify individuals in the base population that possess the desired trait values. These individuals will serve as a starting point for the breeding program and are often referred to as selected parents. After this, we'd have to calculate the selectional difference. So calculate the difference between the mean trait value of the selected parents and the mean trait value of the entire base population. The selection differential S represents how much better the selected parents are in terms of the trait interested compared to the base population. After this, we'd have to calculate our selection intensity. So selection intensity quantifies the strength of selection, which depends on the factors such as how many individuals are selected and how much they deviate from the mean. It can be calculated as, so I is proportion selected. For example, if you select the top 10% of individuals, your I would be 0.10. Now we need to find our R value, which is the selection response. So the R value estimates how much the mean trait value of the selected offspring generation will improve due to the selection. which is calculated as shown below, where we have our equation, where h squared is the heritability, s is the selection differential, and I is the selection intensity. Now we'd have to estimate our h squared. This can be estimated through statistical analysis of phenotypic and genetic data if available. Now we'll have to predict our response to selection. So we'd use the calculated values for h squared, s, and I to predict the response to selection, r. This response represents the expected change in the mean trait value of the selected offspring generation. Now that's all that's left is to implement the selection and evaluate the progress. So we'll select the offspring from the selected parents based on the trait values and continue the breeding program. Over successive generations, we'd monitor the trait's mean value in the selected population. We'd compare it to the mean value in the base population to assess the effectiveness of our selection program and make adjustments as needed.